Our next speaker is John Badalamenti from University of Minnesota.
Okay, thank you, Cesar, for the introduction. So for those of you that don't know, I actually did my PhD here with Cesar and Rosie, who is also in the audience. And today I'm going to be telling you about kind of an extension of some work that I started as a PhD student here, looking at electrodes not only as a tool to study respiration in bacteria, but as a novel microbiological platform for actually enriching these organisms from cool places. Can you guys still hear me? Okay, good. So I know it's a very short time slot, but I do want to take some time just to acknowledge some folks here. Obviously, everyone in the Bond-Negronic labs. I'd like to especially thank the folks at Enrichment for inviting me down to speak, not only today, but to host one of the workshops yesterday, as well as some folks from PacBio and the Minnesota Supercomputing Institute and some of my funding sources. So before I get into some science, I actually want to give everyone a little history lesson. So you wouldn't be crazy if you thought that this looked like a subway map. This is a railway map from the early 20th century of northern Minnesota. You can see this is the far southwestern corner here of Lake Superior. This is Duluth. The Twin Cities are down here. This is Minnesota's Iron Range. To say it is expansive is an understatement. There are estimates placing that the total production of iron from this region of Minnesota from 1900 to 1980 accounts for 60% of all of the iron mined in North America. So it's very likely that some of the steel that we're sitting on right now came from this range. So it's tremendously linked, intricately linked to Minnesota's economy and to Minnesota's history. But why do we care about that? Well, it turns out that located way up here on the northern edge of the Iron Range is a mine called the Sudan Mine. This was the first iron mine in Minnesota, so it's Minnesota's oldest. It's its deepest underground mine. And fortunately for us microbiologists, it was left abandoned and given to the state, donated as a state park. So it's actively maintained. There's running electricity down there. They do tours. And so you can see here, level 27, this is the deepest level of the mine. It's located a half mile underground, 713 meters below the surface. And what's really striking about this mine is these beautiful banded iron formations. This is a massive 2.7 billion year old banded iron formation. And it's microorganisms that are responsible for these banding patterns that we see in the rocks, or at least we think they might be. So one of my hypotheses is that we have this really rich iron mine, and so there might be novel organisms down there that we can access. And the fact that this mine still exists allows us to approach that question. Now, fortunately for us, microbial electrochemists, or I guess me in particular, before the mine was abandoned, they cut some exploratory boreholes through the rock to try and track where the vein of hematite was that they were mining. And so now there's this anoxic ancient brine slowly flowing out of these boreholes. It's 3 times saltier than seawater. It's circumneutral pH. It's high in reduced iron. And it's extremely low biomass. So when I was collecting samples, this is actually concentrated. The biomass was concentrated 100 times with tangential flow filtration. You can still see that it's incredibly clear. This, even after concentration 100-fold, we're talking 10 to the 4th, maybe 10 to the 5th cells per milliliter. So it's incredibly dilute. And so we're asking the question, how can we enrich selectively for organisms that are present at very low relative abundance in this population and very, very low absolute abundance in this population? And I think you can all guess where this is going. So we bring these back to the lab, and we enrich them on poised graphite electrodes under anaerobic conditions. So this is a bank of our new reactor designs that we use in the Bond lab, and ask any of us to tell you why we think they're pretty cool. Okay, so I have to give Daniel some credit here because this is one of Daniel's ideas, and a former postdoc in Jeff and Daniel's lab, Zara Summers, was, I guess, the guinea pig for this experiment. So what we have here, this is the edge of one of the boreholes, again, located on level 27, a half mile underground. This is the brine slowly seeping out from it. And what we decided to do was allow the microorganisms deep down in the borehole to access the high redox potential electron acceptor oxygen in the mine shaft air without actually having to be exposed to it. So we encased a wire in a glass rod and stuck it down this borehole and allowed these microorganisms to do respiration with a very high potential electron acceptor. Zara let that incubate in situ for several months. Then we brought this anode back to the lab. She inoculated it into a bioreactor. And after about 10 days of enrichment, we saw this beautiful exponential growth. She saw that the current responded positively to medium replacement. And so we decided to do some further enrichment and attempt to isolate this. So fast forward about 2 years, Zara left, I took over some of her work, and isolation, of course, is not trivial, but I'll spare you the details. And so we'll fast forward to the punchline. This is Desulforamonas sudanensis, strain WTL for will to live. It has 96% ribosomal RNA gene sequence identity with Desulforamonas michiganensis. And you can see here, we see a nice, I'd call that semi-exponential growth curve. Not very high current densities, but again, this organism in pure culture was able to respire electrodes as it did during the enrichment. Now, what's most striking about this isolate is under the same pH, the same temperature, we see a shift in the cyclic voltammogram about 100 millivolts more positive compared to Geobacter sulfureducens, suggesting that this isolate has redox machinery tuned to capture higher redox potential electron acceptors. So this was really exciting. And everybody loves to push buttons on the potentiostat, and that's great. And I think that can be really informative. But what I really cared about is, in probing the deep subsurface, can we learn anything about the Geobacter ACA and Desulforamonad ACA families to expand our knowledge about extracellular electron transfer, particularly in the subsurface? And so I undertook a genome sequencing effort, and unfortunately it doesn't look that great on this slide, but I will try to walk you through it. This is the complete circular genome of Desulforamonas sudanensis. It's the first ever closed genome for any Desulforamonas isolate. And we've been studying Desulforamonads now for like 40 years, so I thought that was pretty cool. These are all the protein sequences. Any of these dots and triangles here, these are predicted regulatory proteins, two-component signaling cascades, et cetera. Anything in the interior here, these are repeats. These are repetitive DNA sequence, 500 base pairs or greater at 95% identity or greater. If any of you have ever done a de novo assembly with short reads, these are all the places where that assembly would break. And so we actually used PacBio data, and you'll hear me talk more about that later today to close this genome. In red, around the outer perimeter here, are all the multi-heme C-type cytochromes. And I apologize that it's not showing up here on this projector. But up here, we see IMCH and CBCL, the proteins that Daniel talked about yesterday. If you want to know more about CBCL, go see Lori's poster today. And we also see several extracellular conduit clusters. If you want to know more about that, go see Fernanda's poster tomorrow. We see mercury methylation, and we see a tandem duplication of the ribosomal RNA operon, suggesting that we may have actually selected for this in the lab. When we look a little closely at one of the cytochromes, I should mention there are 38 altogether. This one right here has a whopping 69 putative heme binding motifs, which is just astounding. It's the most that we've ever seen in any sequenced multi-heme cytochrome. So we're not really sure why that is, but some really interesting biology going on. And so I want to zoom in on this section of the genome over here for a second, because this is what's really, really cool about this genome, and I could stare at this all day. So these are two C-type cytochromes here, and one of them, which has a predicted 11 heme binding sites, is located within a predicted prophage. So this is viral DNA integrated into the chromosome. And this is the first line of evidence, I think, supporting the hypothesis that I and probably many others have, that the diversity of cytochromes in the environment could be a result of horizontal gene transfer. And so this was really exciting and I think validated our approach in using electrodes to get at really rare members of the subsurface. So to that end, I decided to basically repeat what Zara did. I did it a little more simply, though. I simply went down to the mine, grabbed some brine, and stuck it straight into a bioreactor without enriching in situ. And I went to a different borehole for this. And after about a month of enrichment, we saw this beautiful exponential growth. And we were able to harvest quite a few cells off the electrode. From this, I was able to get about 10 micrograms of DNA. And I want to underscore the electrode enrichment approach because it obviates the need to use iron, which can contaminate your DNA extraction. So if you're interested in sequencing, I think the electrode is a really good opportunity. So if we look at the community before enrichment, not surprisingly, we see a very diverse community, and we don't really see many delta proteobacteria. But as soon as we enrich, we get over 85% delta proteobacteria, and there are two dominant species in this delta proteobacteria slice that I want to focus on. So because we got so much biomass, we were able to do PacBio sequencing. It allowed us to entertain the idea of actually doing PacBio sequencing on a mixed community. Because we had such good results on an isolate, we said, we have a really high enrichment of a couple delta proteobacteria. Can we assemble this out? So we talked to the folks at PacBio. We had some trouble sequencing this library, but we were able to cobble together reads from 2 SMART cells. And even when things were not going our way, this is what we got. This is a complete circular genome of a microorganism that has never grown before in the laboratory. It's the second complete genome of a Desulfuramonas. We call it Desulfuramonas boabicus, after the Ojibwe word for iron. You can see it's incredibly repetitive. This is the most repetitive, to my knowledge, most repetitive genome that's ever been captured from a metagenome. In blue here are all the places where a short-read metagenomic assembly broke, not surprisingly, at a lot of the repeat sequences. Again, we have some really, really striking features in this genome. The first of which, we have IMCH and CBCL located very close to each other on the genome. We have a tandem CRISPR array totaling about 5 kilobase pairs. And we have another tandem duplication of the ribosomal RNA operon. So this really does suggest that we're selecting for this when we bring these organisms into the lab. But again, these aren't the coolest things. But they're pretty cool. This isn't even the coolest. This is what I really want to drive home, and this is why I think long-read metagenomics has to be the way for how we characterize microbial communities on electrodes, because we're not dealing with a high degree of diversity, and we're dealing with situations where we are not limited by biomass in most cases. So this is a repeat, a near-perfect 4 kilobase pair repeat of the C-terminal domain of a multi-heme C-type cytochrome. And you can see here that the Illumina assembly captured one instance of this, but not the other. So what appears to have happened is a duplication of a C-type cytochrome and then an in-frame fusion to 2 different N-terminal, either anchors or signal sequences. And so we have 2 copies of a cytochrome domain fused to 2 different N-terminal regions. And this fundamentally underscores the utility of long-read sequencing in resolving genomic complexity like this. We don't necessarily care about the number of cytochromes. We also care about their arrangement. We care about clusters, conduit clusters. We care about duplications and things like that. So this was really, really exciting. And then finally, these bars here are the individual PacBio reads. You can see just how long they are, covering this region and anchoring it into proper genomic context. So we know that this is real. And then just finally, my last couple slides here, I want to do a comparison of a short read and long read metagenomic assembly. So we had the 3 most abundant organisms in here were Desulfuramonas boabicus, which I just showed you, something that came back nearly 100% identical to Desulfuramonas sudanensis, which I showed you first, and a Marinobacter strain. If we tally up the number of contigs that we got from the short read assembly alone, we're in the several hundreds. They come out in a lot of bits and pieces. And when that happens, you have to do all kinds of crazy computational approaches like ESOM and binning to try to put those genome pieces back together into single bins. With PacBio, we were able to resolve 211 contigs down to 1, 441 contigs down to 1, and 2 orders of magnitude improvement in the number of contigs for this Marinobacter strain here. So it completely eliminated the need to do a binning approach like this and allowed us to quickly fast forward to asking questions about the biology in this system. So if I don't sound like a broken record, I should, because I think long reads are here to stay. And if you haven't heard, PacBio actually just released a new platform earlier this week that is giving 7 times the throughput per SMART cell for half the price of the instrument. So I think that's incredibly exciting. Daniel showed this tree yesterday, and so what these 2 new genomes allow us to do is to step back and look at the phylogeny of the Geobacteriaceae and the Desulfuramonadaceae. And what we see is that the Geobacteriaceae, which are typically isolated from sediment, freshwater environments, cluster down here, and they tend to have more cytochromes than their more halophilic or alkalophilic counterparts. And so these are the two geoalkalibacter isolates that we know about, and the two disulfur-monads that I sequenced cluster out over here with another disulfur-monus that was also enriched on electrodes. You can see here they have about half the cytochrome content of many of the geobacter down here. And so finally, stepping back even further, the whole point of doing this is to ask the question, how much further do we have to sequence? How much further do we have to go before we can start to make sense of the pan-genome of extracellular electron transfer in the Geobacteriaceae? And I think the good news is we're getting close. So we're up to about 25 genomes now, including the 2 that I did. And we're starting to saturate and only add maybe a handful of families for every new genome that we sequence. But we're getting close here. I think we can be cautiously optimistic. But I still think it's a little bit of a mess, because if you look at the core genome, for any random sampling of 5 or more genomes in this family, they all seem to have a different way of tackling the same problem. And so we're able to knock off, say for Geobacter sulfur-reducens, every time we sequence a new isolate, based on homology, we can maybe knock off a couple of families, but there's always going to be something new. And there's always going to be innovation and divergence among the different strains. And so I think this is really exciting, and I think this is a good excuse for me to do more sequencing, which is always fun. So I think I'm near the end here, so I just want to thank everyone for their attention, and I would be happy to take questions.
Hi.
Hi.
If we could have just a quick technical discussion.
Sure.
Sorry, just about, you used PacBio, and you didn't talk about using both, so correcting your reads, your PacBio error rate, correcting it with the short reads, but did you do that or not?
We didn't need to. We had enough PacBio coverage even from 2 poorly loaded smart cells that we were able to do self-correction.
Okay, the figure that you showed there
Yes.
had lots of overlap, but not much uniform overlap. Was that just by coincidence in
Which
the
figure are you referring to?
Well, you had PacBio, you said, you had about like half a dozen reads per part of it was one of your zooms.
Oh, I see. This one?
Like this one, yeah. Do you see what I mean?
This plot cuts off the number of reads mapped to 10, but the total genome coverage for this was over 50x.
Okay.
I just didn't plot all the reads because it looked too cluttered. So
Okay,
I capped that track at 10.
Okay, so you had 50x, so you had enough for the overcome the error rate.
Yep.
Okay, thanks a lot.
Yep. And then I should also mention that because we did Illumina sequencing on that same metagenomic sample, I went back and used the Illumina reads to polish the PacBio assembly even further. So
that was my question.
Yeah, I'm confident in the
So
total base call accuracy.
So you did use the short reads?
Not until the very end, just to scrub out. I think there were 20 indels left in the entire genome.
Okay. And you know about Oxford Nanopore?
Yeah. I think they're going to be a game-changing technology, but I think they have a long way to go before we have access to it.
Well, we have access to it. We're using
I would love to have access to it.
We're using it.
Okay.
Yeah.
Cool.
And it's fun because it's so small. But thanks a lot.
Yep. There's a microphone coming.
You got 2 genome of uncultured candidate. So how about other strain in your enriched consult here?
So no matter how we tried to assemble it, it looked like we had 4 organisms in our electrode enrichment. 2 of them comprised 85% of the community, or closer to 90%. The other one was over 5%. So by the time we got down to the 4th organism in there, which is an alpha proteobacterium that I haven't looked that closely at, we were at low enough coverage where the PacBio assembly is, de novo assembly is not really working. So we need to incorporate some of the short read data with that.
Okay,
Yeah.
I just had a quick question. You, some of the strains were augmented or were magnified with a fuel cell, but then a bunch of them weren't, right?
Mm-hmm.
What does that mean for the ones that weren't in terms of like how they're making how they're surviving down there?
Well, I mean, an electrode is a tremendous selective pressure, right? We're asking the question, who can transfer electrons across a cell membrane? And so there are microorganisms that live in that environment that make a living by doing other metabolism than metal reduction. But we're not asking, when we bring them back to the lab and do a selective enrichment, we're not asking for those bugs to come along for the ride.
I guess that was my question. I mean, there are a lot of other ways of gaining energy out there other than mimicking a fuel cell in those sort of environments?
Right, and so we do have metagenomic samples, short reads only, because it's extremely low biomass, of the native brine. And so we're hoping to use that information to characterize the native communities as well.
Any other questions?
One more.
So your biological source is 2.5 billion ago, right?
Say that again, I'm sorry.
2.5, your brine
Yes.
is 2.5 billion old, right?
Well, I don't know about the brine. The brine is older than the mine itself. I think I can safely say that.
So
but I wouldn't necessarily feel comfortable saying that the brine itself is 2.7 billion years old.
Okay, so as a case, maybe your strain is really close to the origin of life, but still the same level of the C-type cytochromes is existed in that strain. That means maybe the geobacter-like strain has already have the same type of C-type cytochrome at that age. Is it correct? Correctly thinking?
I don't know that I would have enough information to stake that claim.
Yeah, in my feelings, of course, the old strain have a close to ancient, and then separate from our field, our life field. So in that case, maybe separate type of the evolution happens, and maybe new family or new protein family should be accumulated on such kind of old lives. So
yeah, I mean, I could see a scenario where there was a very core set of cytochromes present in the common ancestor to the Geobacteriaceae that then diverged and then recombined, and then they innovated beyond that. But saying that it comes from an old environment and saying that it's a more ancestral organism, I don't know if I would.
How about the other strain?
What's that?
How about the other strains like Clostridia from your original community? You did Illumina sequence and assembly from the original brine community. Maybe those kind of guys has different strains like Clostridia or such kind of guys.
Yeah,
right?
so the native brine communities do have some novel family and order level lineages
So
that we're
those
trying
kind
to
of
bin out.
Yeah, those kind of guys have a new family of the new protein family or known families.
I haven't looked at the data closely enough yet to say, but I'm guessing yes.
Mm-hmm. Interesting. Thank you very much.
We're going to have a break now of 20 minutes. So be back by 3:10. Let's thank Badalamenti.